In humans, pain is a distressing feeling often caused by intense or damaging stimuli. Whether animals apart from humans also experience pain is often contentious despite being scientifically verifiable. The standard measure of pain in humans is how a person reports that pain, for example, on a pain scale. Pain is defined by the International Association for the Study of Pain as an unpleasant sensory and emotional experience associated with actual or potential tissue damage, or described in terms of such damage. Only the person experiencing the pain can know the pain's quality and intensity, and the degree of suffering. However, for non-human animals, it is harder, if even possible, to know whether an emotional experience has occurred. Therefore, this concept is often excluded in definitions of pain in animals, such as that provided by Zimmerman. An aversive sensory experience caused by actual or potential injury that elicits protective motor and vegetative reactions, results in learned avoidance and may modify species-specific behavior, including social behavior. Non-human animals cannot report their feelings to language using humans in the same manner as human communication, but observation of their behavior provides a reasonable indication as to the extent of their pain. Just as with doctors and medics who sometimes share no common language with their patients, the indicators of pain can still be understood. According to the U.S. National Research Council Committee on Recognition and Alleviation of Pain in Laboratory Animals, pain is experienced by many animal species, including mammals and possibly all vertebrates. The experience of pain. Although there are numerous definitions of pain, almost all involve two key components. First, nociception is required. This is the ability to detect noxious stimuli which evoke a reflex response that rapidly moves the entire animal, or the affected part of its body, away from the source of the stimulus. The concept of nociception does not imply any adverse, subjective feeling. It is a reflex action. An example in humans would be the rapid withdrawal of a finger that has touched something hot. The withdrawal occurs before any sensation of pain is actually experienced. The second component is the experience of pain itself, or suffering, the internal, emotional interpretation of the nociceptive experience. Again in humans, this is when the withdrawn finger begins to hurt, moments after the withdrawal. Pain is therefore a private, emotional experience. Pain cannot be directly measured in other animals, including other humans. Responses to putatively painful stimuli can be measured, but not the experience itself. To address this problem when assessing the capacity of other species to experience pain, argument by analogy is used. This is based on the principle that if an animal responds to a stimulus in a similar way to ourselves, it is likely to have had an analogous experience. Reflex response to painful stimuli. Nociception usually involves the transmission of a signal along nerve fibers from the site of a noxious stimulus at the periphery to the spinal cord. Although this signal is also transmitted onto the brain, a reflex response, such as flinching or withdrawal of a limb, is produced by return signals originating in the spinal cord. Thus, both physiological and behavioral responses to nociception can be detected, and no reference need be made to a conscious experience of pain. Based such criteria, nociception has been observed in all major animal taxa. Awareness of pain. Nerve impulses from nociceptors may reach the brain, where information about the stimulus, e.g. quality, location, and intensity, and effect, unpleasantness, are registered. Though the brain activity involved has been studied, the brain processes underlying conscious awareness are not well understood. Adaptive value. The adaptive value of nociception is obvious. An organism detecting a noxious stimulus immediately withdraws the limb, appendage or entire body from the noxious stimulus and thereby avoids further potential injury. However, a characteristic of pain, in mammals at least, is that pain can result in hyperalgesia, a heightened sensitivity to noxious stimuli, and allodynia, a heightened sensitivity to non-noxious stimuli. When this heightened sensitization occurs, the adaptive value is less clear. First, the pain arising from the heightened sensitization can be disproportionate to the actual tissue damage caused. Second, the heightened sensitization may also become chronic, persisting well beyond the tissue's healing. This can mean that rather than the actual tissue damage causing pain, it is the pain due to the heightened sensitization that becomes the concern. This means the sensitization process is sometimes termed maladaptive. It is often suggested hyperalgesia and allodynia assist organisms to protect themselves during healing, but experimental evidence to support this has been lacking. In 2014, the adaptive value of sensitization due to injury was tested using the predatory interactions between longfin inshore squid, Doroteuthis pile, and black sea bass, Centroprastes striata, which are natural predators of this squid. If injured squid are targeted by a base, they began their defensive behaviors sooner, indicated by greater alert distances and longer flight initiation distances, than uninjured squid. If anesthetic 1% ethanol and magnesium chloride is administered prior to the injury, this prevents the sensitization and blocks the behavioral effect. The authors claim this study is the first experimental evidence to support the argument that nociceptive sensitization is actually an adaptive response to injuries. Argument by analogy. To assess the capacity of other species to consciously suffer pain, we resort to argument by analogy. That is, if an animal responds to a stimulus the way a human does, it is likely to have had an analogous experience. If we stick a pin in a chimpanzee's finger and she rapidly withdraws her hand, we use argument by analogy and infer that, like us, she felt pain. It might be argued that consistency requires us infer, also, that a cockroach experiences conscious pain when it writhes after being stuck with a pin. The usual counter-argument is that although the physiology of consciousness is not understood, it clearly involves complex brain processes not present in relatively simple organisms. Other analogies have been pointed out. For example, when given a choice of foods, rats and chickens with clinical symptoms of pain will consume more of an analgesic-containing food than animals not in pain. Additionally, the consumption of the analgesic carprofen in lame chickens was positively correlated to the severity of lameness, and consumption resulted in an improved gait. Such anthropomorphic arguments face the criticism that physical reactions indicating pain may be neither the cause nor result of conscious states, and the approach is subject to criticism of anthropomorphic interpretation. For example, a single-celled organism such as an amoeba may writhe after being exposed to noxious stimuli despite the absence of nociception. History. The idea that animals might not experience pain or suffering as humans do traces back at least to the 17th-century French philosopher, René Descartes, who argued that animals lack consciousness. Researchers remained unsure into the 1980s as to whether animals experience pain, and veterinarians trained in the U.S. before 1989 were simply taught to ignore animal pain. In his interactions with scientists and other veterinarians, Bernard Rollin was regularly asked to prove that animals are conscious, and to provide scientifically acceptable grounds for claiming that they feel pain. Some authors say that the view that animals feel pain differently is now a minority view. Academic reviews of the topic are more equivocal, noting that, although it is likely that some animals have at least simple conscious thoughts and feelings, some authors continue to question how reliably animal mental states can be determined. In different species, the ability to experience pain in an animal, or another human for that matter, cannot be determined directly, but it may be inferred through analogous physiological and behavioral reactions. Although many animals share similar mechanisms of pain detection to those of humans, have similar areas of the brain involved in processing pain, and show similar pain behaviors, it is notoriously difficult to assess how animals actually experience pain. Nociception. Nociceptive nerves, which preferentially detect potential injury-causing stimuli, have been identified in a variety of animals, including invertebrates. The medicinal leech, Hirudo medicinalis, and sea slug are classic model systems for studying nociception. Many other vertebrate and invertebrate animals also show nociceptive reflex responses similar to our own. Pain. Many animals also exhibit more complex behavioral and physiological changes indicative of the ability to experience pain. They eat less food, their normal behavior is disrupted, their social behavior is suppressed, they may adopt unusual behavior patterns, they may emit characteristic distress calls, experience respiratory and cardiovascular changes, as well as inflammation and release of stress hormones. Some criteria that may indicate the potential of another species to feel pain include: has a suitable nervous system and sensory receptors, physiological changes to noxious stimuli, displays protective motor reactions that might include reduced use of an affected area such as limping, rubbing, holding or autotomy, has opioid receptors and shows reduced responses to noxious stimuli when given analgesics and local anesthetics, shows trade-offs between stimulus avoidance and other motivational requirements, shows avoidance learning, high cognitive ability and sentience. Vertebrates. Fish. A typical human cutaneous nerve contains 83% C-type trauma receptors, the type responsible for transmitting signals described by humans as excruciating pain. The same nerves in humans with congenital insensitivity to pain have only 24 to 28% C-type receptors. The rainbow trout has about 5% C-type fibers, while sharks and rays have 0%. Nevertheless, fish have been shown to have sensory neurons that are sensitive to damaging stimuli and are physiologically identical to human nociceptors. Behavioral and physiological responses to a painful event appear comparable to those seen in amphibians, birds, and mammals, and administration of an analgesic drug reduces these responses in fish. Animal welfare advocates have raised concerns about the possible suffering of fish caused by angling. Some countries, e.g., Germany, have banned specific types of fishing, and the British RSPCA now formally prosecutes individuals who are cruel to fish. Invertebrates. Though it has been argued that most invertebrates do not feel pain, there is some evidence that invertebrates, especially the decapod crustaceans, e.g. crabs and lobsters, and cephalopods, e.g. octopuses, exhibit behavioral and physiological reactions indicating they may have the capacity for this experience. Nociceptors have been found in nematodes, annelids and mollusks. Most insects do not possess nociceptors, one known exception being the fruit fly. Invertebrates, endogenous opioids are neurochemicals that moderate pain by interacting with opiate receptors. Opioid peptides and opiate receptors occur naturally in nematodes, mollusks, insects and crustaceans. The presence of opioids in crustaceans has been interpreted as an indication that lobsters may be able to experience pain, although it has been claimed. At present no certain conclusion can be drawn. One suggested reason for rejecting a pain experience in invertebrates is that invertebrate brains are too small. However, brain size does not necessarily equate to complexity of function. Moreover, weight for body weight, the cephalopod brain is in the same size bracket as the vertebrate brain, smaller than that of birds and mammals, but as big as or bigger than most fish brains. Since September 2010, all cephalopods being used for scientific purposes in the EU are protected by EU Directive 2010/63, EU, which states, there is scientific evidence of their cephalopods' ability to experience pain, suffering, distress and lasting harm. In the UK, animal protection legislation means that cephalopods used for scientific purposes must be killed humanely, according to prescribed methods, known as Schedule 1 methods of euthanasia, known to minimize suffering. In medicine and research. Veterinary medicine. Veterinary medicine uses, for actual or potential animal pain, the same analgesics and anesthetics as used in humans. Dolorimetry. Dolorimetry, dolor, Latin, pain, grief, is the measurement of the pain response in animals, including humans. It is practiced occasionally in medicine, as a diagnostic tool, and is regularly used in research into the basic science of pain, and in testing the efficacy of analgesics. Non-human animal pain measurement techniques include the paw pressure test, tail flick test, hot plate test and grimace scales. Laboratory animals. Animals are kept in laboratories for a wide range of reasons, some of which may involve pain, suffering or distress, whilst others, e.g. many of those involved in breeding, will not. The extent to which animal testing causes pain and suffering in laboratory animals is the subject of much debate. Marion Stamp Dawkins defines suffering in laboratory animals as the experience of one of a wide range of extremely unpleasant subjective mental states. The U.S. National Research Council has published guidelines on the care and use of laboratory animals, as well as a report on recognizing and alleviating pain in vertebrates. The United States Department of Agriculture defines a painful procedure in an animal study as one that would reasonably be expected to cause more than slight or momentary pain or distress in a human being to which that procedure was applied. Some critics argue that, paradoxically, researchers raised in the era of increased awareness of animal welfare may be inclined to deny that animals are in pain simply because they do not want to see themselves as people who inflict it. PETA, however, argues that there is no doubt about animals in laboratories being inflicted with pain. In the UK, animal research likely to cause pain, suffering, distress or lasting harm is regulated by the Animals, Scientific Procedures Act 1986 and research with the potential to cause pain is regulated by the Animal Welfare Act of 1966 in the U.S. In the U.S., researchers are not required to provide laboratory animals with pain relief if the administration of such drugs would interfere with their experiment. Laboratory animal veterinarian Larry Carbone writes, without question, present public policy allows humans to cause laboratory animals unalleviated pain. The AWA, the Guide for the Care and Use of Laboratory Animals, and current public health service policy all allow for the conduct of what are often called Category E studies, experiments in which animals are expected to undergo significant pain or distress that will be left untreated because treatments for pain would be expected to interfere with the experiment. Severity scales. 11 countries have national classification systems of pain and suffering experienced by animals used in research: Australia, Canada, Finland, Germany, the Republic of Ireland, the Netherlands, New Zealand, Poland, Sweden, Switzerland, and the UK. The U.S. also has a mandated National Scientific Animal Use Classification System, but it is markedly different from other countries in that it reports on whether pain-relieving drugs were required and/or used. The first severity scales were implemented in 1986 by Finland and the UK. The number of severity categories ranges between 3, Sweden and Finland, and 9, Australia. In the UK, research projects are classified as mild, moderate, and substantial. In terms of the suffering, the researchers conducting the study say they may cause. A fourth category of unclassified means the animal was anesthetized and killed without recovering consciousness. It should be remembered that in the UK system, many research projects, e.g. transgenic breeding, feeding distasteful food, will require a license under the Animals, Scientific Procedures Act 1986, but may cause little or no pain or suffering. In December 2001, 39% 296 of project licenses in force were classified as mild, 55% 811 as moderate, 2% 63 as substantial, and 4% 139 as unclassified. In 2009, of the project licenses issued, 35% 187 were classified as mild, 61% 330 as moderate, 2% 13 as severe, and 2% 11 as unclassified. In the U.S., the Guide for the Care and Use of Laboratory Animals defines the parameters for animal testing regulations. It states, "The ability to experience and respond to pain is widespread in the animal kingdom. Pain is a stressor and, if not relieved, can lead to unacceptable levels of stress and distress in animals." Quote, the guide states that the ability to recognize the symptoms of pain in different species is essential for the people caring for and using animals. Accordingly, all issues of animal pain and distress, and their potential treatment with analgesia and anesthesia, are required regulatory issues for animal protocol approval. See also References